Hello viewers. Today I will discuss on electron transport chain and oxidative phosphorylation. During the catabolism of carbohydrates, fats, proteins, these macromolecules are oxidized into simpler monomeric units. And during the oxidation, the electrons released are captured by electron acceptors like NAD+, FAD+, and sometimes NADP+, and they are converted to NADH, FADH2 and NADPH respectively. Now electrons pass from NADH to several electron acceptors arranged as a chain, and in case of aerobic organisms, oxygen becomes the final electron acceptor. This process is called aerobic respiration. Aerobic respiration occurs in all aerobic microorganisms and in eukaryotes. Now during the electron transfer, the free energy released is utilized to phosphorylate ADP. This phosphorylation is called oxidative phosphorylation, since the energy for phosphorylation comes from oxidation of a molecule. In prokaryotes, oxidative phosphorylation and electron transport chain takes place in the plasma membrane, whereas in eukaryotes, oxidative phosphorylation and electron transport chain takes place in the inner mitochondrial membrane. Another type of respiration is called anaerobic respiration, where an exogenous electron acceptor other than oxygen is the final electron acceptor. Normally, nitrate, sulfate, carbon dioxide, sometimes some metals and organic molecules act as the final electron acceptor. All anaerobic organisms and archaea, they do anaerobic respiration. Now, apart from oxidative phosphorylation, two other phosphorylation systems exist in all living systems. One is called photophosphorylation or photosynthesis, and the other one is substrate level phosphorylation. In photophosphorylation, photosynthetic organisms and plants, they trap the solar energy and they form ATP and NADPH. Using this ATP and NADPH, they synthesize carbohydrate and other organic molecules from environmental carbon dioxide and water molecules. And in substrate level phosphorylation, the energy for phosphorylation comes from a high energy organic compound. When the compound changes its form, the energy released is trapped as phosphorylation of ADP to ATP. Substrate level phosphorylation occurs during respiration, fermentation, etc. and it is the simplest and least evolved way to produce ATP. Now I will go into the detail of electron transport chain and oxidative phosphorylation. Eugene Kennedy and Albert Leninger in 1948 discovered that mitochondria in eukaryotes is the site for electron transport chain and oxidative phosphorylation. Mitochondrion as the gram-negative organisms has two membranes. The outer membrane is permeable to almost all small molecules having molecular weight less than 5000 Dalton, but the inner mitochondrial membrane is impermeable to most of the small molecules. It is only permeable to carbon dioxide, water molecule and oxygen. But there are many transporters present in the inner mitochondrial membrane, and these transporters allow the passage of specific molecules from intermembrane space to the matrix and vice versa. Electron transport chain is composed of a number of enzyme complexes, complex 1, 2, 3 and 4. Each enzyme complex is again composed of several protein subunits and many different cofactors like flavin adenine dinucleotide, flavin mononucleotide and iron sulfur centers. These cofactors help in the transportation of electrons through the complexes. Now complex 1, the first complex of electron transport chain, it is called NADH coenzyme Q oxidoreductase. It is a large enzyme complex composed of 42 different protein subunits, and it has flavin mononucleotide dependent enzyme NADH dehydrogenase and at least 6 iron sulfur centers. And all the electron acceptors are arranged in the increasing order of their reduction potential values. Reduction potential value of an electron acceptor denotes its affinity. The more reduction potential value, the more electron affinity of that electron acceptor. The complex 1 accepts electron from NADH. 2 electrons pass from NADH through FMN center and through the iron sulfur centers. It goes to coenzyme Q. And during the electron transportation, the free energy released, that free energy drives 4 protons from matrix to the intermembrane space. That is, complex 1 acts like a proton pump. Now coenzyme Q, it is a benzoquinone with a long isoprenoid side chain. It is a small organic molecule, so lipid soluble. It can freely diffuse through the inner mitochondrial membrane. Ubiquinone, after accepting 1 electron, is converted to semiquinone, and then accepting another electron, it is converted to quinol. Quinone or ubiquinone transfers electrons from complex 1 to complex 3. Now complex 2. Complex 2 is also called succinate dehydrogenase. This enzyme complex is involved in TCA cycle. It converts succinate to fumarate. It is also a large complex and composed of 4 different protein subunits and FAD as its cofactor. Electrons from succinate are first donated to FAD. From there, through iron sulfur center, it goes to quinone or coenzyme Q. Electrons released during the beta oxidation of fatty acyl CoA also enter into the electron transport chain via this complex 2. So coenzyme Q transports electrons from complex 1 to complex 3 and from complex 2 to complex 3. Complex 3 is called ubiquinol cytochrome C oxidoreductase. This complex is composed of, it is a dimer of 2 monomers each having 11 subunits, and the active center is composed of 1 molecule of cytochrome B, 1 risque iron sulfur center and cytochrome C1. What happens here? 1 molecule of ubiquinol donates 1 electron to cytochrome C1 and to a Q present in the matrix side, making it a semiquinone. A second ubiquinol molecule then donates 1 electron to cytochrome C and 1 electron to the semiquinone, making it a quinol. So from 2 molecules of ubiquinol, we get back 1 ubiquinol and 2 electrons are released to cytochrome C1 one by one. And ubiquinol is converted to in its oxidized form. It is called the Q cycle. From cytochrome C1, electrons go to cytochrome C molecules. Cytochrome C molecules are heme-containing proteins that can carry 1 electron at a time. So 2 electrons are carried to complex 4 by 2 cytochrome C molecules. The fourth complex is called cytochrome oxidase. It is also made of 13 subunits. Subunit 1 contains 2 heme groups A and A3 and a copper center called copper B. And subunit 2 carries copper A center. Electron from cytochrome C enters into the complex 4. First it goes to copper A center, then to heme A3 and then to copper B center. And finally, in case of aerobic respiration, oxygen is the final electron acceptor. After accepting the electron, oxygen combines with protons from matrix and it is converted into water molecule. And during the electron transportation, 2 protons are transported from matrix to intermembrane space. Hence, the transfer of 2 electrons from NADH to O2 through the respiratory chain can be written as this. And if we calculate the standard free energy change using the standard reduction potential values, then we see For each pair of electron transfer from NADH to O2, about 220 kilojoule of energy is produced per mole of NADH. Due to the proton transport from matrix to intermembrane space, proton concentration increases in the intermembrane space. This increased proton concentration gives rise to number 1, a chemical potential energy. Why? Because due to the concentration variation of a chemical species H+ in two regions separated by a membrane. And number 2, an electrical potential energy due to the movement of a positive ion from matrix to intermembrane space without a counter ion. And together this electrochemical energy is stored in this concentration gradient called PMF or proton motive force. Now, how this proton gradient drive the synthesis of ATP? To explain this question, Peter Michel proposed a model called chemi-osmotic model. According to this model, when the protons passively flow back into the matrix from the intermembrane space, protons enter through a ATP synthase and the PMF drives the synthesis of ATP. Michel used the word chemi-osmotic to describe an enzymatic reaction which involves at the same time a chemical process and a transport process. Now, ATP synthesis occurs by with the help of an enzyme complex called ATP synthase or complex 5. It is present in the inner mitochondrial membrane in case of eukaryotes. ATP synthase has two components, FO, which is the integral membrane protein part, and F1, the peripheral membrane part. FO, this fragment is sensitive to the antibiotic oligomycin and hence it is called FO. F1 is composed of 9 different protein subunits of 5 types having a composition alpha 3, beta 3, gamma, delta and epsilon. The delta subunit joins the FO to F1 with the help of epsilon, and each beta subunit carries a catalytic site for ATP synthesis where ADP and PI can bind and phosphorylation occurs. Now, to describe how ATP synthase produces ATP, Paul Boyer proposed a mechanism called rotational catalysis mechanism. According to this mechanism, each beta subunit captures ADP and PI from the surrounding, and as 3 protons come back from intermembrane space to matrix, the rotation occurs through the alpha beta spheroid. ADP and PI combines to form an ATP molecule, and 3 beta subunits interact in such a way that at any point of time, if one beta subunit takes beta empty conformation, its neighbor to one side must take the beta ADP conformation, that means where ADP and PI remains bound, and the other neighbor must be present in its beta ATP conformation, that means where ATP has just formed. And one complete rotation, that means 360-degree rotation of the gamma subunit causes the synthesis of 3 ATP molecules with the combined with the entry of 9 protons from intermembrane space to matrix. So, to synthesize ATP, ADP and PI must enter into the matrix and the newly formed ATP molecules must exit the matrix. But as we know that the inner mitochondrial membrane is impermeable to most of the molecules, this work is done by 2 translocase proteins. One is adenine nucleotide translocase and the other one is phosphate translocase. Adenine nucleotide translocase is an antiporter and it allows the passage of one ADP molecule from cytosol or intermembrane space to matrix, at the same time an ATP molecule from matrix to the intermembrane space. And phosphate translocase, it allows the transportation of one phosphate inorganic phosphate molecule and one proton into the matrix. It is a symporter. So, to produce or to synthesize one ATP molecule, 4 protons must enter into the matrix from the intermembrane space. So we have seen that to synthesize one molecule of ATP, 4 protons must enter into the matrix from the intermembrane space. And in the electron transport chain, we have seen that for one pair of electron transportation through electron transport chain, 10 protons move to the intermembrane space from matrix. So for per electron pair transfer through electron transport chain, 2.5 molecules of ATP are generated when one pair of electron is transferred from NADH to O2. And for one electron pair transfer from FADH2 to O2, since 6 protons go to the intermembrane space from matrix, 1.5 molecules of ATP are generated. Now, in eukaryotes, electron transport chain occurs in the inner mitochondrial membrane, and the NADH dehydrogenase, it is capable to use the NADH molecules present in the mitochondrial matrix only. So, the NADH molecules produced during glycolysis in the cytosol must enter into the matrix. In eukaryotes, 2 shuttle systems carry the cytosolic equivalent of NADH into the mitochondrial matrix. One is called malate aspartate shuttle. It is the most active shuttle system for NADH, and it is present in liver, kidney and heart. What happens here? The cytosolic NADH, the reducing equivalent of cytosolic NADH is donated to cytosolic oxaloacetate, which is converted to malate by cytosolic malate dehydrogenase enzyme. Now, on the inner mitochondrial membrane, there is a transporter called malate alpha-ketoglutarate transporter. This transporter allows the movement of malate from cytosol to the matrix. In the matrix, malate donates the reducing equivalent to NAD+, and the NADH form can easily donate the electron to the electron transport chain. But the oxaloacetate converted into malate in the cytosol must be regenerated. So, another transporter called glutamate aspartate transporter helps to continue the cycle. And the cells which use this shuttle can generate 2.5 molecules of ATP from one electron pair transfer from NADH to O2. Another shuttle system that is operative in skeletal muscle and brain cell mitochondria is called glycerol 3-phosphate shuttle. Here the reducing equivalent from NADH is given to flavin adenine dinucleotide and then to coenzyme Q. That means it acts like the complex 2 to coenzyme Q pathway. So, 1.5 molecules of ATP are generated for the NADH molecules that come to the electron transport chain via this shuttle. So, if we look into the total ATP molecule generation, then from 1 mole of glucose, about 32 or 30 moles of ATP are generated depending on the tissue. That means depending on the shuttle system the cells have used. In prokaryotes, electron transport chain and oxidative phosphorylation occurs in the plasma membrane. All aerobic microorganisms, protists, fungi, they follow this pathway. The electron transport chain, the mechanism of electron transport chain and oxidative phosphorylation is similar in prokaryotes and eukaryotes. But the electron transport chain is more complicated in prokaryotes compared to the eukaryotic system, because there are several different electron donors and several different electron acceptors. Electron can enter into the electron transport chain of a prokaryote at the level of a dehydrogenase enzyme, at the level of quinone pool, and also at the level of cytochrome electron carrier. A simplified version of electron transport chain in E. coli has been shown here. E. coli has developed 2 different branches of electron transport chain depending on the oxygen level. When oxygen level is high, they follow the BO branch, and more protons move into the periplasmic space from the cytosol, and more ATP is generated and cells grow very rapidly. But when oxygen level is low, they follow the BD pathway, and less protons move from the cytosol to the periplasmic space and their growth becomes slow. And the PMF, proton motive force generated along the plasma membrane, is utilized for the active transport of molecules needed by the cell. Number 2, for the motility of the bacterium, and number 3, for the synthesis of ATP by ATP synthase. This is the structure of E. coli ATP synthase. It is made of 8 different subunits and it is the simplest ATP synthase known. So today we have learned how prokaryotes and eukaryotes carry on electron transport chain and oxidative phosphorylation, and how oxidative phosphorylation is dependent on the electron transport chain. Without the electron transport chain, there will be no oxidative phosphorylation or synthesis of ATP.